Bismillahirrahmanirrahim. Assalamu alaikum students. Today our topic is excretion in animals. We will see how waste products are excreted out from the bodies of human beings, animals and unicellular organisms. The first category is invertebrates and vertebrates. So you know what are invertebrates? The types of organisms which do not have backbone in them. And vertebrates are the opposite of invertebrates, which contains a backbone or a vertebral column in their bodies. So students, basically you should know that the excretory product in the body of animals is nitrogen wastes. For example, when we are sweating, many toxic chemicals like excess salts and electrolytes will be removed out from the body, which are harmful for the body. There are three different nitrogenous wastes which are nitrogenous end products, and they are removed out of the body in three different ways. We have urea, we have ammonia, and we have uric acid, which are the three end products of the nitrogenous waste basically. So during the metabolism, a number of toxic materials are produced which are actually the byproducts of different chemical reactions, particularly the nitrogen containing compounds, as I have already told you. Now, excretion is the removal of chemical wastes from the body which are produced by the metabolic processes within the cells. And the excretion ends in the elimination of the excretory products which I have told you are ammonia, urea and uric acid. Now we will see what is the relationship between excretory products and the habitats of different animals which excrete ammonia, urea and uric acid. The exact nature of the excretory product is determined mainly by the availability of water. So the production of ammonia, urea and uric acid basically depends upon how much water is available in that particular environment. So you can see ammonia is produced by the organisms which are aquatic, means which are found in the water bodies. Urea is produced both in aquatic and terrestrial environment, whereas uric acid is merely produced by the organisms of the terrestrial environment. Now if we talk about ammonia, it is a highly toxic substance out of all the three because it tends to raise the pH of the body fluids and it interferes with the membrane transport functions. It is highly soluble in water, so it needs a high amount of water. For example, 500 ml of water is required to eliminate 1 gram of nitrogen in the form of ammonia. So it diffuses rapidly across the cell membrane because it highly becomes dissolved into the water. Therefore, it is excreted out rapidly. Such plenty of water can only be afforded obviously by the aquatic organisms, particularly those which live in the fresh water. Because you know, in the previous topic we talked about the freshwater organisms and the marine organisms, and we also discussed that fresh water is diluted water which contains less amount of salts and solutes. For example, the protozoans that we discussed in the previous topic, sponges, some fishes and the coelentrates. These are the groups of organisms that we discuss in the previous topic in the previous year's book. Now, ammonia is wasted as the major nitrogenous waste product, which is called ammonotelic organisms. Next nitrogenous waste product is urea. So urea is less toxic as compared to the ammonia, and organisms with less fresh water available, such as some marine organisms which live in less water content, and all the terrestrial organisms remove most of the nitrogenous waste in the form of urea, because 50 ml of water will be required to eliminate 1 gram of nitrogenous waste, that is urea. And urea is 100,000 times less toxic than ammonia. So animals which excrete urea as their major nitrogenous waste products are called ureotelic. So this can be asked in the short question, that what is the difference between ammonotelic and ureotelic organisms, and the third that we will discuss in the next slide, that can also be asked in the short question. Uric acid is the purine which is even less toxic than urea. You know, purines, pyrimidines, hypoxanthine, xanthine, uric acid, allantoin, all these are the excretory products which are excreted out in a large quantity from the body of living organisms, because purines and pyrimidines, you know, they contain nitrogen in them. But creatinine, creatine and TMAO that we discussed in the previous topic, these are removed out from the bodies in a little quantity. So this is your short question that can be asked in the paper. Now 1 gram of nitrogen requires only 1 ml of water to be excreted out. And the organisms which excrete uric acid, they are called uricotelic. Usually birds are uricotelic, which excrete uric acid in the form of small crystals, and it has evolved in two groups with major water loss problems, especially the terrestrial invertebrates and egg-laying vertebrates. So in the diagram you can see the amino acids and the nitrogenous bases. Ammonia, urea and uric acid, they contain the amine groups, that is NH2, most aquatic animals including most bony fishes, they excrete out large amounts of dilute urine, and ammonia is released from their bodies. All the mammals, amphibians and some bony fishes, they excrete urea. Birds and many reptiles and insects and snails, they release uric acid. Now the excretory system of vertebrates, or the excretory system of man. The excretory system of man consists of kidneys, ureter, urinary bladder and a tubular urethra. You can see in the picture the two kidneys, which are bean shaped, are present here. Attached with the kidneys are two tubes which are called ureters. Then ends at the bladder, that is a bag-shaped structure which empties into the urethra. The location of the kidneys is that they are located against the dorsal abdominal wall, and the excretory system or the kidneys, they lie on either side of the vertebral column, starting between 12th thoracic and 3rd lumbar vertebrae. Thoracic region is the chest region that we discussed in the previous chapter. The right kidney is little lower than the left kidney, and the difference is about 0.5 centimeter. The total weights of the kidneys are less than 1% of the total body weight, while it receives 20% of the blood supplied with each cardiac beat. So the ureters, which are the long tubes measuring 25 centimeter length, they begins or starts from the funnel shaped renal pelvis. Renal pelvis is not very clear in the diagram, but the ureters extends out from the funnel shaped structures called the renal pelvis out from the concave sides of the kidneys. Now urinary bladder, that is a muscular organ, it is hollow from inside and its function is to store time, and it is present in the pelvic region. Pelvic region is the hip region, and as I have told you, it serves as a urine reservoir. Now the urethra. This is a tube that carries urine from the urinary bladder to the outside of the body. The urethra empties near the vagina in the females or through the penis in the males. So it is present outside from the body in males. Then if we talk about these structures in detail, the structure and functions of the kidney is called nephrology. Nephrology is the science that is concerned with the structure, functions and diseases of the kidneys. So everything that we study under the heading of kidneys is called nephrology. This word originates from the unit structures present inside the kidneys, which are called the nephrons. Kidneys are reddish brown, bean-shaped structures, and they are covered with fibrous connective tissue layer, which is called a capsule. And at the other side of the kidneys are present the adrenal glands. Now you can see the cross section of a kidney in detail. The structure of the kidney consists of two regions basically. The outer darker region is called cortex, and the inner pale region is called the medulla. Now the other structure is hilus. Hilus is, if we see at the lateral surface of each kidney, it will be convex, but the medial side will be deeply concave. The entrance to this sinus is termed as hilus. Hilus enters into the kidney through the small structures which are called sinuses, and they are called renal sinuses because they are related to the kidneys. So, the depression or the invagination at the concave side of the kidney through which blood vessels and ureter enters the kidney is called a hilus. Now, if we talk about the internal structure of the kidney, that is called renal medulla. It contains the cone-shaped structures which are called the renal pyramids. These renal pyramids contain millions of structures which are called nephrons. Renal papillae. The base of each pyramid is called the renal papillae, which is pointed towards the center of the kidney. So here you can see these conical pointed regions of the renal pyramids are called renal papillae. Now there will be always a blood supply. There is a vein and an artery that carries the oxygenated blood and that carries away the deoxygenated blood from the kidney. And minor calyces and major calyces. The extensions of the renal papillae are called calyces. The smaller ones are called minor calyces, which empties into the larger ones, which are called major calyces, and they finally empties into the hilus, and hilus empties through the renal pelvis into the ureters. Now the structure of nephron, which is the unit cell, unit structure of the kidneys present in the pyramids of the kidney. A nephron is a very complicated structure and it has a great importance. Nephron consists of renal capsule and renal tubules. Now if we talk about the renal capsule, it consists of two parts. One is called glomerulus and the other is called Bowman's capsule. Glomerular capsule is surrounded by the Bowman's capsule, which is a cup-shaped structure. If we talk about the glomerulus capsule, it is composed of a network of capillaries. And if we talk about the Bowman's capsule, it just surrounds the glomerulus capsule. It is an expansion at the closed end of the renal tubule. Now moving forward, the tubules which extends out from the glomerular capsule are proximal convoluted tubule and distal convoluted tubule. Proximal one is the tubule that leads away from the Bowman's capsule and becomes highly coiled. This coiled portion of the tubules is called proximal convoluted tubule. Another structure that is present at the base of the whole nephron is called loop of Henle. Loop of Henle contains the descending and ascending limb. Now you can see the darker, thicker tubes which are surrounded by the capillary network. They are called ascending, which are going upward, and descending, which are going downward limbs. And collectively they are called the loop of Henle or the loop of the nephron. Then the loop of Henle also contains a structure that is called vasa recta. Vasa recta is an additional capillary network that extends down to form a loop of vessels. Okay, next will be the distal convoluted tubule. The ascending limb returns to the region of the renal capsule where it becomes highly coiled again, and this is called the distal convoluted tubule. Proximal is something that is near and distal is something that is away. So both these tubules which are present near and away from the Bowman's capsule are called proximal and distal respectively. Now there comes an enlarged broad tube that is called the collecting duct. It contains or it receives many nephrons. Many collecting ducts combine together to form larger collecting ducts, which first empties into the minor calyces and then they empties into the major calyces through an opening in the renal papillae. So we have already talked about the papillae as well. Now if we talk about these capillaries, we will discuss them one by one. Blood circulation to the nephron is through renal arteries, interlobular arteries, efferent arterioles, peritubular capillaries, vasa recta, and interlobular veins and the renal veins. Now the renal arteries, they supply the kidneys with a pair of renal arteries from the aorta, and aorta is the major capillary that is connected to the heart, and it will supply the kidneys with the oxygenated blood. Now the interlobular arteries, renal artery basically is divided up and it forms different branches which are called interlobular arteries. The most important are efferent arteriole and efferent arteriole. Efferent arterioles supply blood to the glomerular capillaries of the renal capsule, and efferent arterioles, whose diameter is somewhat less than that of the efferent vessel, each efferent arteriole exits from the glomerulus. So the function of efferent arteriole is to supply the glomerulus with blood, and efferent will take away the blood from the glomerulus. Now the peritubular capillaries, after each efferent arteriole exits or leaves the glomerulus, it gives rise to a plexus of capillaries. Plexus means a complicated network of capillaries which are called the peritubular capillaries, and they will surround the proximal and distal tubules. Interlobular veins. So the function of interlobular veins will be just opposite to the function of the interlobular arteries. And the renal vein exits the kidney and connects it to the inferior vena cava. Inferior vena cava is the major vein of the body which carries the deoxygenated blood back to the heart. Thank you so much.